So we are 20 participants. So I will open the webinar. So thank you very much for attending this monthly EUROTOX webinar. So today it will be Dr. Costanza Rovida, she is going to give us a talk about regulatory status of skin sensitization and alternative methods. So Costanza is a chemist by education from University of Milan, and she's now project manager at Team Mastery, which is a dedicated REACH consulting company. She's listed at the European List of Registered Toxicology, the ERT. She's also collaborating as a scientific officer with the Center of Alternative of Animal Tests, the CAT, which has one part in Europe and one part in the US, and the CAT has been at the origin of the alternative method. It is a pioneer for the development of alternative method in toxicology. And Costanza is also the actual president of ECOPA. So I don't know if you know about ECOPA. It's a European platform for alternative to animal tests. That's so there is national platform and in international, which is regrouping in ECOPA, which is the official European platform. And she's a very well-known and recognized expert in regulatory affairs and animal-free methods in toxicology. And she gives numerous presentations in international meetings such as EUROTOX and ESTIV and other meetings. So we are very pleased to host today Costanza. And thank you again, Costanza, for accepting to give this webinar. And I'm sure all the people would benefit of it. So please, for all the attendees, please shut down your microphone. The session is recorded. And please put your question in the chat. It will be a 45-minute talk, then 15 minutes question. So it's always better to have the question in the chat before, and then I can read the question. And after, if you have more questions during the question and answer part, you can do it, of course. Okay. So Costanza, the floor is yours. Thank you again.
Okay. Thank you very much, Mark. Very nice introduction. Maybe too much. I'll try to do my best to share with you talking about the work of the regulatory affairs specialist. And what we do, what I do is to do what is requested by the legislation to market the new chemical product, but by keeping an eye to waiving a new animal test. That's because of my involvement with CAT and ECOPA. And also because I strongly believe that this is the future for a very, not only from ethical point of view, but also for the scientific reason, because I really trust in vitro methods. So first of all, how to implement the regulatory needs. There are some defined steps. So I must define the aim of what I need, locate where you need it, identify the relevant legislations, identify the authority, find out the guidelines, define the best strategy for implementation and actions. Be careful that there are always new amendments and new laws. So it's a work that you can never relax. So you always have to keep yourself updated. So let's start from the beginning and aim of what I need, and then I jump immediately to the very last point. You see that action is in the end. So before performing a new test, you have to go through all those steps. And please don't do what is usually what I often see, that people just perform the test and then call me, so please interpret the results. No, because it's much better and much more convenient, because in some cases, some tests that are useless are performed. So it's much better if the analysis of the problem is performed before. And then locate where you need, because the legislations are different from country to country. Today, we will talk about the European Union. But what I say is more or less applicable to other regions for classification and labeling. Actually, CLP is implementing the GHS system, so the Global Harmonized System, that harmonizes the definition of the classification rules in a way that a chemical as standard hazard phrase and pictograms. In theory, all over the world now, the GHS system is implemented more or less everywhere. And in the EU, there are also nice guidance on the application of the CLP criteria, where all the methods that are accepted are listed and explained. If we go to into Annex 1 of CLP, there is a definition of what respiratory or skin sensitization is. And look that respiratory sensitization is refers to hypersensitivity of the airways occurring after inhalation, while skin sensitization is allergic response after skin contact. So be careful of this difference, because I had problems, and respiratory sensitization is not referring to allergic reaction. It's hypersensitivity is different, and then also that it's linked to the inhalation exposure or skin contact. So it means that if I have a chemical that is inducing asthma by skin contact, it's still skin sensitized. And these are related to the hazard phrases. We have H334 for respiratory sensitization, H317 for skin. And today we will talk only about the skin sensitization, because respiratory sensitization is completely different. And when we have a substance which is classified as skin sensitizer, there are also thresholds that are defining the possibility that a mixture containing that substance is classified as skin sensitizer. And these are 1% for a generic category 1 and 0.1% if the category is 1A. Be careful that these are generic concentration limits, so they are applicable when there is no additional information. So it can be that specific concentration limits can be lower than that. And also there are other limitations, for example, if a chemical is used for cosmetic purpose. And but how we can define the two subcategories, 1A and 1B? Unfortunately, this is linked to results from animal tests or evidence in humans. So in theory, it means that if we just apply the CLP criteria, in vitro methods are not allowed. And so in theory, my talk would be finished, but unfortunately, it's not, luckily, it's not like that, because if we move to the GHS, in the 10th revision of the 27th of July last year, we see that the possibility to add in chemical in vitro methods and also non-test methods, so QSR and modeling, are embedded in the tiered approach. And there is also this very nice specification, because it's and or. So it means that the alternative approach is fully acceptable. And so this part of the GHS has not been implemented into a CLP yet, but hopefully it will be soon. I don't know why, because you know that the activity in the EU is more or less frozen at the moment, waiting for the new election next month. So let's move now, because CLP is just for classification, but let's see what's inside specific regulation, like the 1107 for plant protection products, or REACH, or the regulation on biocidal products or cosmetics. So if we start from REACH, REACH Annex 7 is about the substances that are marketed in quantities above 1 ton per year. So it's the very first one. So it means the skin sensitization has to be assessed for all chemicals that are registered in REACH. So this is the copy and paste of the text of the consolidated version of the regulation. It means that it contains all the different amendments. REACH is published in 2006. So until now, there are many changes. So and this one regarding skin sensitization was published in 2017. So it means that the possibility to apply alternative method, in vitro methods, has been implemented since 2017. And what is the exact text? It says that if we want to assess skin sensitization, we have to first try to apply in vitro and in chemical test methods, referring to the ABSC, so molecular interaction with skin protein, inflammatory response in keratinocytes, and activator on the dendritic cells. We will see them because they are the three main group of methods that are applicable. And what's more, it says that this first in vitro step is mandatory. So you have to first try to use the in vitro and move to in vivo only if the in vitro approach is not applicable for some reasons. That's very important. I can tell you that if you submit now a dossier with the in vivo methods and nothing else, no explanation, it will be rejected by the authority. That's REACH. If we move to BPR, so the regulation on biocidal products, we have Annex 2 and 3 in this regulation. Annex 2 is about active substances, while Annex 3 is about products containing the biocide active substance. And the skin sensitization text is more or less the same. It's not as detailed as REACH, but it's still accepting and referring to in vitro testing. And here we don't have the phrase that in vitro test must be performed before, but still it is part of the text. This amendment, this accepting sensitization is from 2021, so some years later than REACH. And unfortunately, if we move to plant protection product, this hasn't been happened yet. So for PPPs, we have two regulations, 203 and 204 of 2013. One about active substances and the latter about products. And this text still contains reference to in vivo methods only. So does this mean that if we have to deal with the PPP, we have to perform in vivo methods? No, because the next step of my ballot list is the identify the authority. So we have in the EU we have EFSA and ECHA. ECHA for BPR and REACH, EFSA for PPPs and other things. And in this case, especially when you are asking the authorization for a new plant protection product, the dialogue with the authority is very, very close. And in that case, you can still ask for the permission to do in vitro instead of in vivo, and I can tell you that is always acceptable. So that's the end. Let's move now to what is the guidelines that we have to apply to perform this assessment. So the main reference to guidelines comes from the OECD, the Organization for Economic and Cooperation and Development. The OECD there are now 40 participants all over the world. The blue are those that are participating. And what does it mean that we are using? So the OECD is publishing the test guidelines that are applicable to all those countries. And what does it mean? That, first of all, those guidelines are accepted internationally as standard method. So if I do a skin sense test in the EU, because I have to comply for REACH, that test is accepted also in Australia, in the US, or in Canada, because there is this there is this mutual acceptance of data, so that's very important. Another very important point is that guidelines are used by professionals in the industry, academia, and government, so they are used by anyone, also for research purposes, if you like. And these guidelines are continuously expanded and updated. I must tell you that the working group at the OECD are very much active and are very always they pay a lot of attention of what's coming, and so that they are very the guidelines are quite well updated. And remember that you always have to apply the good laboratory practice. If you perform the best methods, but it's not in GLP, it can be rejected. So regarding skin sense, of course, we have to look at section 4 on health effect. OECD is publishing guidelines for all type of assessment. And in addition to the test guidelines, there are also another section regarding series on testing and assessment, which is explaining additional information like validation reports, guidance, how to deal with difficult substances, and so on. So we will see the something about skin sense also in this area. So regarding the OECD test guideline for skin sense, we have some from in vivo. The Gainia peak maximization test and bullet test in the 406, which is historical, the very first method that was used for skin sensitization. Then we have the Lacon-Lino assay and modification of the original methods that's validated. And then we have the 3 validated in vitro methods, and we will talk about them in more details now. What's the principle behind those 3 methods? The principle is that of the adverse outcome pathway. The adverse outcome pathway is a principle that states that whatever is happening in our organism, it's the consequence of a sequence of events, starting from of course, the exposure of the chemical, and then there is the molecular initiating event, so the first interact of our substance with some receptor within our organism. This molecular initiating event is triggering a sequence of other events that in the end may lead to some adverse event in the organism. Regarding skin, we have the first link to some proteins. So we have a covalent binding between chemicals and our proteins. This is triggering keratinocyte activation and dendritic cell activation. Dendritic cells activated are moving towards the lymphocytes and where they can provoke a T cell activation and proliferation. And this is the very beginning of the allergic process. When, in case the T cells is reacting in this way, it may happen that when there is a second exposure to my chemicals, to the chemicals that we are analyzing, the lymphocytes are reacting, and they can provoke a skin sensitization. So this is the principle. So we have all those steps with a number of key events, and the methods reflect those schemes. So if we concentrate on the final adverse outcome on the organism, we see that we have to measure a real allergic contact dermatitis. And this is what's happening when a human, a person is sensitized. So if I'm sure that many of you experience allergic reactions, and maybe they have performed the test in the hospital, so that they put usually in the back or on the arms your substance to measure the reaction. So it means that you are allergic. So it means that you're measuring the adverse outcome. And regarding the in vivo test, there is the OECD 406 that is measuring in Guinea pig with those two methods, GPNT and Euler test, how many animals react to a chemical. So it's a real allergic reaction. If we focus on the T cell proliferation only, that's exactly the endpoint measured by the local linode assay. And before, we have the 3 in vitro method. So we have we will see them, some methods that are measuring the protein binding, others that are measuring the keratinocyte activation, and others that are measuring the dendritic cell activation. There are 3 steps with 3 different methods that require an additional analysis, and that's the defined approach described in the OECD 497, to understand and to combine the results to get to a conclusion. So let's start from the first one. That's the OECD 442C, that's about the adverse outcome pathway. And it's based on in chemical, it's called a chemical analysis, chemical methods, because it's an HPLC. So the principle is that a substance that is potentially become a skin sensitizer, first of all, does a covalent binding with proteins with ending with lysine or cysteine. And so by measuring this binding, I can have an idea of the possibility of that chemical to be a sensitizer. So the method is a depletion. So I start by measuring the presence of cysteine and lysine, and then I measure how much of them are disappearing, let's see like that, from my HPLC trace. It's called in chemical because there is no living tissue or cells involved. In the test guidance, there are 3 methods: the direct peptide activity assay, the amino acid derivative reactivity assay, and the kinetic direct reactivity assay. So the differences are that, okay, the DIPRA and the ADRA are just measuring the binding, while the last one is the kinetic, so the velocity of this reaction may occur, and it can give you a very good clue on the potency of the chemical. So first of all, you decide whether your substance is or is not a skin sensitizer, and then you measure the kinetic. This approach is fantastic. It's very predictive, very reproducible. Again, the KDPRA can give a nice clue on the potency of the substance. And there is also the possibility to distinguish between skin and respiratory sensitizer, because it has been demonstrated that respiratory sensitizer have a higher affinity with lysine, but this is not really validated yet. Unfortunately, there are also limitations because there are some chemicals that interact with keratinocyte and dendritic cell directly without this previous binding with the protein, and those are basically metals. There are some limited applicability with UVCBs, in particular when UVCBs are unknown and variable composition complex reaction products or biological materials, so are very complex substances like herbal extract or many chemicals are UVCBs. And the limited applicability is in particular when there are big differences between the solubility of the different components of the UVCBs. And solubility is also another issue because it's a chemical reaction, so you need to have your substance fully dissolved and fully dissolved in water, basically. And there are only few solvents that are accepted, like acetonitrile, isopropanol. So are those solvents that are compatible with the HPLC analysis. So, for example, the MSO is not ideal. it's a chemical test method, so of course there is no metabolism, and stability has to be also assessed, because it's not only the stability of the chemicals, but there are some chemicals that may oxidize cysteine, for example, and so cysteine is depleted even if there is no binding. So to apply this method, you really have to think of the chemistry of your substance, of the possible reaction, and see whether it's applicable or not. To decide whether a chemical is or is not a sensitizer, there are tables that is considering the ratio between the depletion, so the area of the HPLC analysis where the reaction has occurred, and the area of the reference control. This is always an average between several repetitions, so it's very stable. For the other, the thresholds are different. I take this opportunity to add something else. Remember that when there are methods that seem to be equivalent, because more or less the principle is the same, like in this case, remember to always look at the prediction model, so the threshold that are defined to decide whether your chemical is or is not, in this case, sensitizer, in other case can be with other properties. The second key event is described in OECD 442D. It's about keratinocyte activation. This test method was updated in 2022. And it's measuring the inflammatory response of the keratinocyte by activation of the KEAP1 on the antioxidant response element. So the principle is that my chemical is covalent binding to the cysteine and to modifying the protein KEAP1. That is, as a consequence, has this dissociation of the NRF2, and the NRF2 is activating this antioxidant response element that is linked to a luciferase. So it means that there is the emission of light, and this is measured. This measure is very, very easy, and it's easy to measure, so it's also a very reproducible method. It's based on cells that, of course, have been modified to incorporate the luciferase gene. And again, it's very reproducible. It's also giving a clue on potency. But again, there is the problems of solubility, and only it's the opposite of the other one. Only the MSO is acceptable. In this case, there is the possibility to accept also the dispersion, because as soon as there is interaction with the cells, the method is acceptable. Limited metabolism. Remember that for the DIPRA it was no metabolism. Here it's limited because we are dealing with cells, so in any case there are some metabolism. So in many cases the lack of metabolism is considered the main limitation of in vitro methods. I don't fully agree, because cells have some metabolism, and there are also ways to overcome this limitation. So regarding the key event number 3, it's described in the OECD 442D, and it's about the activation of dendritic cells. In this method, there are 4 methods. 4 methods that are on 3 cell. So we have the H-CLAT, the human cell line activation test, that is based on THP1. And THP1 there are all cell lines. And with the THP1, there is the measure of some cell surface marker linked to dendritic cell maturation, and those are CD86 and CD54. Then we have USense, that it's the principle is the same, but it's using U937, and it's measuring just the CD86. Then we have IL-8 leukocyte with the L8 RNA and the gas skin. So all of them have been validated, all of them are accepted. I personally prefer the H-CLAT or the gas skin for two reasons. One is that the H-CLAT is part of the defined approach. We will see it later. And also because both of them are measuring more than one endpoint. So H-CLAT is CD86, CD54, and the gas skin is even based on 196 genes, so it's more complex. And this is important when there are some doubt about the reliability of the result, and to balance in case there are, in some cases you have results that are on the edge of the threshold, and so it's not clear which is the best decision to take. So just little more details on the H-CLAT. Again, the strength is very sensitive. It's applicable to measure gluon potency. The limitations are solubility. The guidelines is asking to be careful if your substance has the log kappa-V of higher than 3.5. I added here a question mark because, in my opinion, it's not a matter of higher octanol water partitioning, but it's the matter of water solubility. So when the substance is not very much soluble in water, and if it has a octanol water partition so high, it means that it's more or less lipophilic. And so you have to be careful. So what does it mean? That it's not that you cannot measure a substance with that KOV, but is that if it's positive, it's acceptable. Of course, if it's negative, you have to be more careful, because you don't know whether it was a real negative or it's just that there was no physical interaction between your chemicals and the cells. That's other limitations. I am mentioning here the fluorescence interference with feature, which is the reagent that is added to the antibody to measure the CD86 and CD54. But in all methods that you are dealing with, you first of all have to consider any possible interference of your chemicals with the test method. So those are the thresholds that are defined for positive results. So it's 150% of for CD86 and 200% for CD54. With cell viability, that's 50%, but of course I prefer a higher value. The cell viability is also very important, because if in a preliminary test we can measure that my chemicals is causing cytotoxicity, it means that even if it's not well soluble, even if it's not a suspension or whatever, it means that it has an effect on the cells. So it means that there is interaction. And in a way, this is a very strong element to decide whether your final results is reliable or not. And for potency, it's considered the minimal induction threshold. So between those two values, it is taken the lowest. Very easy. Regarding the gas skin is on MUS3 cells. And it's interesting because it's measuring 196 gene expression level. And those are measured and assessed with the patent algorithm. So it's interesting because those genes are routed to highlight different conditions like irritation and so on. But the limitation is that it's a patent system. So it means that if you want to implement this method in your lab, you always have to refer to the patent program to analyze the genes. Compared to the HCLAT and to the other methods, there are two other positive things to consider, and that's that has been demonstrated to be applicable also to lab lipophilic substances. So the gas skin has no limitation on the octane water partitioning level. And as it is very sensitive, also the minimum solubility is very low, much lower than the HCLAT. This sensogen can be also included in a higher tier strategy, I would say. So there is the gas potency test to define whether the chemicals is 1A or 1B. And then there is also the GAD air for respiratory sensitizer. This is very interesting. There are many publications on this, but remember that it's not validated. So you can use it in weight of evidence, but not as a standalone method. Another interesting method that's available is the senses. In theory, I like the senses very much because it is based on reconstructed epidermis. So in theory, it includes all three endpoints, the three key events in one method. So, but it's very sensitive. Again, it is based on gene expression, and so it's quite reliable. Unfortunately, the validation was stopped because of some practical problems during the evaluation of the numbers of the results that have been submitted for validation. So the validation of this method is halted. I really hope that it will go on soon. So after this very quick presentation of the available methods, we go to the last point before deciding which action to take, and that's about the best strategy. So I have described three key events, and then how can we combine that? We have other OECD guidelines. We have the 168, which is explaining very well the principle of the adverse outcome pathway and the precise meaning of the methods that have been validated. And then we have last year the guideline 497 about defined approaches. And this is very important because it gives you the possibility to combine the results and present a conclusion to the authority that is fully compliant with the regulation. Remember that there are many other interesting OECD guidelines on skin sensitization, and in particular, I'm referring to the long list of documents that are reporting the results of the validation. There are really many with so many numbers and details that there is no time now to go through them. But I invite you to refer to those documents in case you are analyzing something and you have problems, because when you go into the validation, you may understand better whether there are some details of your protocol or there are some questions related to some specific class of chemicals. So it can be interesting to better refine the conclusion. So This guideline has 3 approaches, I would say. The very famous 2 out of 3 defined approach. So it means that if I have a chemical, I analyze with all 3 methods, and in case there are discrepancies between the results, I just take the 2. So I have the
Trae un cuchillo, por favor. ¿Por qué? Lo tengo.
If there is direct peptide activity assay which is negative, but I have keratinosense and HCAP which are positive, of course I classify the substance as skin sensitizer. And in the guidelines there are also many examples. I'm not very fond of this approach because I'm a chemist, so I don't like to accept the 2 out of 3 as a black box. I want to understand why, in case one of the 3 is not the same as the other 2. And so I want to understand whether there are problems in the protocol or in the chemistry or interferences with some chemicals. So I want to be sure about my conclusion instead of taking the 2 out of 3 as a black box. And actually, we usually prefer the other 2, and that's also it's applying only the direct peptide activity assay and the HCLAP added to a non-test method. And this is very important because it gives you, so before doing a test, we always do modeling. So it's very important to fully understand the chemistry of my chemicals, to understand whether there are functional groups, there are some other methods, other chemicals that are similar, where maybe there are some epidemiological human data. So it's much better. And then you perform the experiment only when it is to confirm a precise hypothesis. So in my opinion, this is a much more robust approach to risk assessment. And in this guidance, there is also explanation on how to determine the category of your skin sensitizer. It's based on the MIT. Do you remember MIT is minimal induction threshold? So it's the minimum concentration that is triggering the classification as skin sensitizer in the HCLAT, plus other ranges for the DBRA, and with numbers. So in the end, you add all the numbers, and so you have to you get to the total battery score that defines the potency associated with that battery. As I was saying before, never forget read across. So never analyze a substance by its own, but always analyze it by considering all available information that you may have on that substance. Anyway, for the definition of these guidelines on defined approach, there was some numbers to understand the sensitivity and specificity. You see that the numbers are very high for the this combined approach. They are higher than the local linear assay. You may be surprised that specificity is so low. You see 44% to 22% for local linear assay, but that's only statistical reasons, because sensitivity is measuring true positive, while specificity is measuring negative. And as in the validation panel of chemicals, the number of negative chemicals are lower than those that are positive. So that's the reason why it's low, but the approach is quite reliable. Just to be sure that you are familiar with those numbers, I am reporting here how they are evaluated. So you have to validate your method with a panel of chemicals where your properties is well known. And for the skin sensitization, it's easier than with other endpoints because there are many humans that are sensitized. And so when we say that the chemical is positive, in many cases we are sure because there are workers that are sensitized to that chemicals, unfortunately. And so you see that specificity is the ratio between true negatives about compared to the number of all negative chemicals. Sensitivities are true positive compared to the number of all positive chemicals. And we have accuracy that is combining, or balanced accuracy that is more or less the same. Remember that we are talking about toxicity. Toxicity is not black and white or red and gray as is in this picture. So there is very small differences. So unless it's clear sensitizer or it's clear non-sensitizer, there is a wide area in between that's not so easy to define whether a chemical is a sensitizer or a non-sensitizer. So I just collect all the numbers, and to define the threshold, I put a level. And of course, if I move the threshold, I can have a higher specificity or higher sensitivity. It depends whether I want to be sure that my method is really highlighting all possible sensitizer, including many false positive or the opposite. Last but not least, remember that to well report all you are doing. I've seen some dossiers that have been rejected by the authority, very well defined from the experimental point of view, all very nice, but the results were not properly reported to the authority, and of course the authority has to reject it. So be very, very precise in this step. You must define the endpoint, the purpose, underline rationale, everything. Don't forget to add a section on known uncertainties. And so I get to conclusion, just to summarize what I've just said. So start from the regulatory framework. Never consider in vivo data as gold reference, because I didn't say that very well, but for example, the local Lino Dessay also has a limitation. There is no unique instruction. You always have to tailor your approach to your own specific method. And the substance should be performed globally. So, okay, I'm defining skin sensitization, but I have to consider water solubility, irritation, many other things. So always consider FISCAM properties. Be careful of the applicability domain before performing your method. Be careful that your method is applicable, and in the OECD guidelines, in the introduction, it's always very well specified. And then interpret all available results. Of course, I didn't mention that, but it's also important because I can perform hundreds of tests, but then I have to consider the cost and the simplicity in afterwards when I have to combine all those results. And with this, I'm finished. Grazie. Thank you for your attention. And I'm here to accept all of your questions.
Thank you very much, Costanza, for this very, very complete overview of skin sensitization, including risk assessment. So we have some questions in the chat. Maybe Andrea, if you don't mind to read your question or to say your question, because it's quite long. So it's two questions, actually, very interesting question. Do you mind asking your question directly with the microphone, or do you want me to read it? Andrea?
Can you hear me?
Yes.
Yes.
Yes. Thanks for the question that I can say you. So the first one is the when you talk about the ratio of lysine and cysteine, 1:10 and 1:50, is considering a large amount, a large excess of the test chemical, right?
No. it's very well defined in the. I don't remember now by heart, but if you read at the guideline, you have to do a precise, a very precise concentration.
Yes, but 1 it's the cysteine and 10 is the test chemical in the ratio, right?
Yeah, the question
Yes.
is which one is in excess, the peptide or the chemicals?
Yes.
Good question. No, usually it's 1-to-1. But I mean, I don't remember. I never considered that, but it's very well defined in the guideline.
Okay.
But
And
why are you asking it? What's your concern?
because when I tried to apply this guideline for 2C, I had this doubt about how to prepare the test chemical solution for the incubation, and maybe I can try to write a little more, but just to this doubt, and okay, I thought maybe this is this is a good case to ask. But
okay, consider that something. There are many things that
Yeah,
I didn't have the opportunity to say, but one of them is that all those methods have a list of performance standards. So they have a list of substance with the let's say very consolidated result. So before performing a test in the lab, you have to analyze those substances to see whether what you are doing is equivalent to what is published in the guideline. And in case you have different results, it means that there is something that is not working well. And and you have to stick on the concentrations that are described in the test guideline.
Okay, thanks.
Yeah, Andrea, the concentration in the guideline are well defined, but it's all, if I remember, it is the chemicals in excess, if I remember.
Okay, thanks for the clarification. So, and also another things that I didn't understand very well is the difference between the reference control A and the reference control B.
In the deep in the deep
Yeah.
I don't remember. I have to look at it, and if you send me an email, I will answer you in private.
Okay, thanks. Can I say also the last question?
Yes.
Thanks. I have to test a chemical mixture of different compound because I have a product with an active ingredient and other stabilizer, for example, to calculate the correct molarity of the test chemical. I tried to consider the percentage of all components and make a mean weight by the relative percentage according to, of course, the molecular weight of each component. Is the correct way to act, to operate, or not?
Yes, it is the correct way, because you know that there is this possibility in case you cannot, you don't know the molecular weight and you cannot have the defined molarity of the concentration. You can use weight, but in this case, you are not considering enough the components that are present in lower concentration. So what you are doing is very good.
Okay.
There is now actually precise instruction on how to work. I have just read, there is a publication very recent on this issue. Again, if you send me an email, I can share you the link, but what you are doing is definitely correct.
Okay, thanks for the answers.
I have a question for Yana Nunik. Which from in silico methods would you recommend, direct or QSR toolbox?
We use the QSR toolbox because the QSR toolbox is also, I mean, when we are assessing a chemical, usually it's not just for skin sense. We do everything. And so with the toolbox is the first approach very comprehensive, but Derek is also very valuable. And actually, there are also other methods. Those are the two that are listed in the guidelines, but there are many other methods that are quite reliable, and you can use it unless you are sure of what you're doing. For example, we are dealing with dyes, and we are building our own QSR, because dyes are in many cases out of the applicability domain of all QSRs. So we are with the university trying to build our own.
Yeah, I have a question on the guard, because there is now an option in 442D. So it's the guard skin now is accepted. And what about the Sensis test? What is that? The regulatory status of Sensis right now?
It's halted. If you go, you know that there is the SAR, the website where there is the validation status of all the methods. And if you go to Sensis, there is a stopped, I think, or halted, I don't remember. And I know that they had practical problems in presenting the numbers for the validation, because validation is a very complex statistical analysis of all the results that you have acquired on a number of methods. And it seems that, if I'm not wrong, there was a mismatch between two submissions, but some really practical problems that invalidated the submission. So they have to redo it from scratch. But that's not because the method is not, from a scientific point of view, unreliable. It's really regarding the validation process. And I don't know when the new submission, you know that the validation is on a voluntary basis, so, and it's very expensive and not easy to promote unless we will have additional funding from the European Commission. We are asking for it in general. We'll see.
There is any more question? I have more questions, but any more question from the people? No. Okay, so I have because there were a question also on gluten allergy in infant, but I think it is not. There is a question about gluten allergy in infant, but I don't think it is.
Oh, by the way, gluten is something else, first of all, and then it's completely different from food allergies. So the immunological process is completely different, so you cannot transpose the results from skin to food allergies. Completely different.
Okay, and so also one more question from my side is that I can see that in the Ashclad assay, so you can calculate a potency, okay, with the MIT, and so now we can use the MIT in a defined approach. That's right.
Yes.
Okay. Okay, I didn't know that, because I thought the Ashclad was only here to detect danger. It was not used for what we call, well, it could be a little bit of risk assessment, but okay. So you can calculate this MIT based on the CD80 signature.
Not by itself, in combination with the DBRA or in combination with the normal. But again, if it's clear, so strong, so evident, of course, you can use it as a standalone method. But most of the cases is always in the, you know, that I've seen, that I have shown the graph with colors. So if it's red, white, okay, but there are many chemicals that are in between. So you have to balance and weight the different results that you obtain with the test methods. Okay,
and I've got one more question. So you don't like the 2 out of 3 approach? That's right. Can you elaborate a little bit more on it, please?
Yeah, because I mean, if I have a keratinosense which is negative and an H-class which is positive, I want to understand why. And so that's why I try to to go into the more detail in depth of the possible interferences of my chemicals with the reagents, if there are some possible problems of during the experiment, or very important, the ratio between your positive results and the cytotoxicity, which is a very good sign of how much your chemicals is interacting with your cells, and many other factors.
Okay. So that's
The stability of your chemical in the medium.
of course. Yeah, because sometimes people think that 2 out of 3 is very simple, but it's very important to know about the assays and what are the pitfalls and the issues with the assays to really interpret it correctly. So, yeah.
Unfortunately, what I see is that DPRA is fantastic, but the applicability is sometimes limited. And so that's another reason why
the solubility is always an issue. Yes.
Yeah.
Okay. There is more question. No, there is.
And that's why I'm waiting for the census, because the census would solve all these problems.
Yeah. It's a nice assay. It has been here for quite a long time. It is still not validated. So I see a lot of thank you very much, nice talk, etc. in the chat, but no more question. And for me, I think I have no more questions also, and it's past 1 o'clock. So again, Costanza, thank you very much for your very nice presentation. And
thank you. It was nice to be with you.
A lot of details, and I hope you answer all the questions. I mean, I'm sure you can contact Costanza if you want, if you have more questions. And remember that the presentation has been recorded and it will be on the YouTube channel of EUROTOX. Okay. So thank you very much again to everybody. Have a very nice day, and thank you again, Costanza, for the nice presentation. Thank you.
Thank you. Have a good day. Bye.
Bye.